Hello, everyone. Thank you for the many people online joining us, and thank you for the people attending in person. This is a special seminar for us because it is a joint seminar bringing together the MLC's very distinguished Chaucer Club, which is dedicated for discussing topics, contemporary topics in cognitive neuroscience, and also the closeNIT seminar, which brings together a community all over the UK that is interested in topics relating to neurotechnologies, and particularly how we can form a feedback loop between technology and humans. And it was really hard for me to come up with a speaker that I knew could fill up both the room here and the virtual room out there. And I thought very hard who would be my absolute favorite speaker in the world. And I actually came up with Sliman Ben Smaya, which is a very dear friend of Juan and I, that very unfortunately passed away for unknown circumstances a few months ago. So these gigantic shoes had to be filled in. So I asked for my second best, guest. And I know Juan shares my love and appreciation of Sliman's work. So I know for him it's a compliment rather than a diss. So now I'm going to say why Juan is, well, now that Sliman is passed away, why Juan is my best choice. Juan is one of the few people I know in the community that has the kind of the triple whammy. So he has really strong, interesting, innovative theoretical framework. And in order to explore these frameworks, he develops his own analytical tools that are then shared by the entire community. And he's been pioneering on a lot of the methodology that is currently just gold standard in the community. And to bridge together these methods with the theories, he does this really, really complex, intricate, challenging experimental work at a level that we don't really see today very often. So for these three reasons, Juan is absolutely one of my favorite scientists in the whole world right now. And considering how young he is, I can only begin to express how excited I am about the work that would come. Is the paper published yet? Can we?
No,
Okay.
it's in press.
Okay, are we allowed to mention it? Yeah, so the paper we would present today is currently in press for Nature, and I think it captures some of the pioneering and exciting work that he's going to show today. So please join me in inviting Juan to the podium.
Thank you, Tamar. I mean, I don't know. I think you're all going to be disappointed after such an introduction. Disappointed, and also apologies because I'm a bit sick, so I may, not be at my best today. But it's a great pleasure for me to be here and to see so many cognitive people caring about movement. Actually, it's really nice. Yeah, and also those online. But like Tamar, I wanted to start saying a few words about Sliman. Sliman was. A very good friend. Actually, for the first time in a long time, I wrote something for a talk. So Sliman was a larger-than-life, unique guy, and as well as a passionate, talented, and creative scientist. And one thing about Sliman is that he really saw beauty in science, and so he thought that science was worth pursuing because it's beautiful, like art. And actually, Sliman was also a really good musician. If you follow him on Twitter or on the social network before known as Twitter, sorry, you will see him posting videos of him playing piano with his children. And actually his bio was neuroscientist, funketeer, husband, and father. And yeah, Sliman was overall, to me, like a force for good in the community. He supported lots of young scientists. He was also like a really good person to talk with because he was always very opinionated and critical. But you could also like really convince him and persuade him if you were right. And I don't know if I was right many times, but I certainly learned a lot, from my conversations with him. And yeah, another thing that I wanted to say is that, again, if you were on Twitter or if you know people who knew Sliman or you knew him, what was most impressive is that everyone was talking about the sadness of losing him as a mentor and a colleague, more so than a scientist, even though he was one of the people who has actually changed how we think about the somatosensory system and has like made, the current like revolution in brain-computer interfaces possible. And, if you want to read like at least one paper after my seminar, I would encourage you to read this paper by Sneeman, Restoring the Sense of Touch with a Prosthetic Hand. This is PNAS 2013. And here Sliman designed an astonishingly beautiful experiment in monkeys to prove that he could stimulate selectively primary somatosensory cortex, and the monkeys could feel being touched in the finger, different fingers with different intensities. And this is what actually later on he and other colleagues translated to humans. So he's Sliman, I guess, like touching the hand of a paralyzed individual. And yeah, this is a really beautiful paper. You should check it out. Another paper that I think is a good testament of Sliman's work is this recent paper where he and Lee Miller went through years and years of experiments to record from the cuneiform nucleus as the sensory entryway to the brain. And, took a lot of talent and creativity to actually be able to record from this and start to understand how sensory information is transformed along the neural axis. So yeah. Anyway, and the last thing I wanted to say is like I will miss Sliman. And I'm sad that we're supposed to have dinner next week together. So I guess it will happen later. Anyway, after this bit of a somber note, I guess I should talk about work and research. So I wanted to start every time that I give a talk to a cognitive audience or not a motor audience, I try to justify why I care about movement. And I would want to say that, regardless of whether you're into music like Sliman or arts or sports or just, hanging out with friends and sharing a meal, movement is how we do all these things that bring us joy and happiness, right? And also we are all neuroscientists here. And the thing is like the brain actually evolved to move. And we know this from studying like this kind of sea polyps, or we think so because of these sea polyps called ascidia, whose life can be summarized as they are born. They use their brain to find like a nice rock that they like. They latch onto it, they digest their own brain, and they live happy and brainlessly ever after. This is, by the way, Daniel Wolpert, a very famous and influential motor neuroscientist, compares this to the academic career, where you use your brain to get to a chair, and then you can imagine what happens next. Yeah. So, like, just a bit more about what we do. So we are interested in movement. We are at Imperial, and what we do, as Tamar says, is combine experiments now in mice with brain recordings, data analysis techniques, and computational models. And I will be talking about all these things. And now we're started experiments in humans. And this is me and my PhD students being excited that I was in the lab and that they could stab my muscles with intramuscular electrodes. But yeah, basically all the work that we do is revolves around a theoretical framework that we proposed a few years ago. We try to bring front and center this idea that single neurons in the brain actually that were long supposed to be the basic computational units of the brain, right? Like actually, it's not like they can, for example, cause movement by modulating their activity independently. So if you think of each of these circles as a neuron, it's not like when a monkey tries to reach, for example, each neuron can do whatever they want to make the monkey move, right? And we know this is the case because biophysical factors like circuit connectivity patterns, common inputs, neuromodulators, all these basically biophysical constraints make neurons work together in specific patterns, right? In patterns of commodulation that I'm trying to represent here in green and blue. And one thing that I would like to stress is that we think it's not like we have one group of neurons that always work together and another group of neurons that always work together and so forth. These patterns involve many neurons, and many neurons can form part of many of these patterns. So our proposal is that the brain does the doing by modulating these population activity patterns that are shaped by these biophysical constraints, and we call these neural modes. And so far, you can complain that I've been only philosophizing, but I want to tell you how you can use this idea to make testable predictions and run experiments and try to learn about the brain. And I should also say that if I don't make sense, please raise your hand and stop me. It's more interesting and more fun than hearing myself for an hour. So all good? Okay. So now imagine that you're recording from 3 neurons using intracortical electrodes like we like doing. So since we're interested in the coordinated activity of these neurons, what we're going to do instead of the classical, I plot their activity for different conditions, right? What I'm going to do is build a space in which each axis is the activity of one neuron. In this case, the firing rate. So now you can imagine that, for example, when a monkey is about to move, the state of these neurons becomes a point in this state space, right? And as the monkey moves, the activity of these neurons changes and will describe some trajectory in this space. Okay. And one thing that we've observed, we meaning the royal we, the community, is that during laboratory tasks from locus to humans, from primary somatosensory regions to prefrontal cortex, if you plot all these trajectories during a task, as I will do later, they remain confined to a small part of this space. So it's not like these trajectories explore the entire space, but they remain confined to a small part of it, which is what you would expect if all these factors that I was talking about constrain population activity, right? So our proposal is that we can understand the brain, and I should say that we call this surface a neural manifold. Maybe you've come across this word. Have you? Anyone? A few nodding people. Okay. And the activity within it and this manifold is defined by the neural modes, and the activity within it we tend to call it latent dynamics. Okay. So what I'm going to talk about today is how we can use this idea of manifolds and latent dynamics to answer to try and shed light onto some questions in systems neuroscience. But one more thing before I do that, it's like, how do neurons fit in this view, right? Because we also know that from many classic experiments that we can learn a lot from single neurons, right? There's place cells and grid cells, one an overprice. And in the motor system, we can see neurons that tell us a lot about behavior. So in this framework, it can be shown mathematically that each neuron is basically a projection of this population dynamics. So you can think about a neuron as, if my hand is the population dynamics, then one neuron would be like one shadow of this population dynamics somewhere. Another neuron would be a shadow somewhere else, and so forth. Okay? So each of these neurons will give you some view of this true population dynamics that we propose to do doing. Does this make sense to everyone? Any questions? Okay. So the two things that I want to tell you about today, one is this thing that Tamar kind of, I guess, like scoop, is that how different animals with unique brains perform the same behavior. The second study, since we are, in the cognitive community, I wanted to talk about how animals plan movements with uncertain goals, which is an ongoing study. And there may be a secret bonus thing in one slide if we have time. But again, I would rather just like make it, answer all your questions along the way. So let's start with experiments, because we also do experiments. So I want you all to close your eyes, please, and touch your nose with your right index finger. And leave your finger there, open your eyes, look around. So you've all, of course, done this. You can rest, right? So this is something that obviously we can all do. Congratulations, and didn't see anyone missing like this guy. But this is something that we all take for granted. But the fact is that I think is quite remarkable, right? Because each of our brains is unique, and even each of our hemispheres is unique, right? So how come that we can all do these simple things, right? So, and as I say, this is a study led by Mostafa Safai and Joanna Chang, a postdoc and a PhD student in the lab, and in collaboration with many people, foremost Matt Berry. So our idea was very simple. Like, we know that the behavioral repertoire of an animal is shaped by evolution over long time scales, and that this process basically culminates in in a genome that basically encodes the instructions to build a brain of a certain class. So what you can see there very clearly is a collection of monkey brains, as you can tell. So, the monkey genome basically has instructions to build monkey brains. And each of these brains is unique, right? But we also know that unique in the cellular composition. But we also know that the way that neurons are connected as microcircuits within layers, within regions, is preserved across all of us, right? We have the same brain regions and the connectivity motifs are preserved. This makes sense, right? And it's kind of trivial. So our hypothesis was that since we believe that these neural manifolds reflect these circuit connectivity constraints, then it's reasonable to assume, we think, that this genome generates instructions to build basically a landscape where the neural manifolds from a species will be built. Okay, so our proposal is that all monkeys are born with the ability to generate the same neural landscape. Okay, and again, this is philosophizing so far. How can we test this empirically? So the way to do it, we did it is as follows. Imagine now that I have two monkeys, each with their unique brain, and they're performing the same behavior. Right? If we are right, and each of these two unique brains instantiates this same common neural landscape, then what follows is that if I record neural activity from these two monkeys, and I look at the latent dynamics within these manifolds, they should be the same. Okay, and what I mean the same is that I should be able, you can look at, imagine that my left hand is the blue trajectories and my right hand is the green trajectories, right? I'm looking at these two hands from different points of view. No, so if I want to reveal that they are the same, all I have to do is like find a way to look at my right and left hand to reveal that they are the same. No, so the way to test our hypothesis is if I record neural activity from two monkeys doing the same task, can I just find like a kind of rotation and a scaling that will reveal that these dynamics are the same? Okay, any questions? Okay, so, and of course, from this prediction follows that if the behavior, if a monkey is doing this and another monkey is doing this, the dynamics will not be as similar, right? So let me walk you through, let me try to convince you that this is the case. So I'll be talking about the most classic motor task, which is instructed delay reaching task. So here we have monkeys basically are using this manipulandum, to move the yellow cursor to the target to get a reward. Okay. And monkeys become very good at this. Here you can see the position of the hand along the X and Y axis as the monkeys are doing this. And these are a few single trials. And the arrow indicates where the target is. And you can see that these trajectories are very similar, right? Because the monkeys are doing the same movements. If we look at the neural activity, what you can see here are rasters of a bunch of neurons from these two different monkeys in primary motor cortex. So the main motor output region of the brain. And these rasters look very different, but this is not surprising because these are different brains, right? And different implants. If I look at the activity within their manifolds, and this is actual data, here you can see the latent trajectories as the monkey C was reaching to each of these targets. So you can see there's kind of curved trajectories, and if you pay attention to the colors and the targets that are in that circle, there's some correspondence. So yellow is next to orange and is next to green and so forth. Okay, so there's some structure. If I show you these trajectories for monkey M, they look completely different, although these common features are still there. Okay. So now the question is, can I take these two sets of trajectories, like I told you before, and basically do the trick, right? This left hand is monkey C, right hand is monkey M. Can I find a rotation that reveals that they are the same? And the answer is, I can. Okay, and this is just like a few single trials. Yes, there's a question over there.
Do all the movements start from the same position?
Yes.
The trajectory.
The trajectories in neural space, you mean?
Yes.
They don't need to because it depends on the neurons you're looking at. So this is space in a way the origin of the space, and actually the geometric details depends on the neurons you're looking at. It's like if you are looking at an object from here or from here. Some topological properties of the bottle, if I show you a more interesting object than a bottle, like if you look at the bottle from here or from here, right, to different points of view, you have the same geometrical properties, but what you are seeing is different. So it's kind of the same idea because you're using different observers to look at the same object.
But you're recording both the same neurons in all conditions. for each one.
Yes. Oh, you mean like within an animal, like all the points not start in the same place? Yes, and I will get to why that is the case later on. But remind me if I jump through it. Yeah. Any other questions?
So do you do only rotations or the non-linear transformations as well you need to do?
We only do.
Mind repeating the question?
Yeah, good point. Thank you. Yeah, the question was whether we only do linear rotations or we do nonlinear transformations. We are only doing linear operations. So we are only rotating and scaling, and you cannot shear. So what the method we're using for those interested is canonical correlation analysis, and we use other simple linear methods to make the point. You can use a more complicated nonlinear method to improve the results, but we wanted to start with the simplest method to make the point. Okay. Okay, I'll continue. So this is just like, squiggly lines. So obviously we need to quantify this, right? So what we did is compute the correlations for each dimension of the manifold. So we consider the manifold has 10 dimensions, and we computed the correlation in these trajectories for each dimension for all trials together, concatenated, and so forth. And you can see that the first 4 or 5 correlations are extremely high for neuroscience, like above 0.6, 0.7, right? But again, this is an associate that this decreases because of how the method works. Again, this is not very meaningful. We need context, right? Context could be a lower bound control that is just like alignment different epochs. I don't think that is interesting. I think the interesting check is asking how similar are these latent dynamics across monkeys compared to how similar they can be. And the most stringent control is how similar are they when I look at the same neurons from the same monkey on the same day performing this task, right? So we just took like 2 sets of trials from the same session and did the same analysis. And what we got was this gray line. So you can see that the latent dynamics are almost as similar across 2 monkeys, the red line, as they can be based on this within animal within day control in gray. And this is one example. This is for 126 comparisons across 3 or 4 monkeys. So I hope this convinces you that these monkeys, as they move, generate very similar latent dynamics. But there's a caveat to this analysis, which is that it doesn't tell me anything about whether these aligned latent dynamics mean anything for behavior, right? Because I've only been looking at the neural activity without caring about the behavior. So what we did to answer that question, or to show that that is the case, was like basically build models, decoders, to predict the movement of the hand. So here you can see the position of the hand again on the X and Y axis during 10 trials from a monkey. And, it's well known in the community that you can build good models to predict within day from the same neurons. So in gray, you can see that our predictions are pretty good. This is, again, same model, same monkey, same day. But the cool thing is that if I build a model in monkey 1 and I test it in monkey 2, it works equally well. So we can predict how a monkey is moving based on a model trained on a different monkey, if we do our alignment. Because if we don't do our alignment, you can see in the gray traces that the decoders are very bad. And to put some numbers, so this is variance explained, you can see that within animal, we normally get like 80%, and across animal, we're getting the same number, roughly, for this comparison, and this was the case for all comparisons. So this means that these aligned latent dynamics can tell us how different monkeys move in their hand. Okay. Yes?
Was this alignment calculated across monkeys on the same day? Does the alignment change every day?
Yeah, there's a question if the alignment changes over day. It changes because normally the neurons we record from are different on different days. So again, you have the same object and you're looking at it from different points of view. And actually, our previous paper, we did this alignment. We developed this alignment to address this question. How can you find a constant model over years? Okay. Any other questions? Okay, I'll move on. Yeah, so is this only a feature of monkeys? So, and let me push this a bit more. So we also were curious to see what happened in mouse motor cortex to see if it will happen in all the species. So we are collaborating a lot with Josh Darman and his former postdoc John Park, who are at Janilia, or used to be in the case of Junsha. And so we started looking at this mouse motor cortical data, but it also has an interesting advantage for our interest, which is that, we could again get like tens of neurons, and you can see that the behavior in the mice is less similar than it was across monkeys. And this is because these were mice reaching and grasping and pulling on a lever, which is not very natural for a mouse compared to a monkey. And also, because these mice were trained for a few weeks, whereas our monkeys had been like, playing this game for many weeks or months or years. So then the consequence of that is like, this is the correlation in the hand movements between mice. So it's like around 0.6 compared to between monkeys, which is very close to 1. So this allowed us to start testing our hypothesis that the similarity in latent dynamics relates to the similarity of the behavior. Because we would expect that mice have less similar dynamics, less similar behavior, so they should have less similar dynamics, right? And this was indeed the case. You can see that the across animal correlations are farther away from the within animal correlations, but they're still pretty large compared to the lower bound control. We can look at this more directly simply by plotting the correlation in behavior versus the correlation in neural activity for each comparison. So what you will see here is that, especially for the mice where there's a broad range of behavioral variability, the more similar the behavior, the more correlated the latent dynamics. Okay. So this kind of supports the notion that our ability to align dynamics is constrained by the similarity of the behavior. Okay. But I bet, like, who is a skeptic in the room? I feel like a lot of you. So then the question is, I'm also a skeptic, so I take it as a compliment. So we wanted to push this even further, also thanks to the help of a reviewer. So if it's in this room, thank you. So, this is kind of like boring tasks where there's a lot of structure, right? It's monkeys doing this from the center, or mice doing this or this from the center, right? So what happens in a more challenging behavior? So we have data from monkeys playing whack-a-mole, the game where basically there's these moles and you have to hammer them very quickly. So where whack-a-mole task was basically there was like a target that popped out and the monkey had to acquire it, and then another one will appear, another one, another one, another one. So they had to do 4 targets very quickly like this in order to get a fruit reward, a juice reward. So they made lots of mistakes, which is good for this. And also they did a lot of movements. These are the hand movements from the top, from 2 different monkeys. Okay. So this looks very messy, but Juan actually went through the kind of significant amount of work of developing a way to match these movements between the 2 monkeys. And you can see a few examples of that. So for example, if you focus on the green traces at the bottom, you can see a dashed line is a movement performed by monkey N, and the solid line is the closest movement performed by monkey C. So you can find like pretty similar movements, but these were less similar than for the previous task. And the cool thing is that we could again align the dynamics. The red trace is still well above the control, but as expected, it's farther away from the upper bound set by the same monkey. Okay. Yes.
Well, quick basic clarification question. So what is the control?
Yeah. So the lower bound control, we did a lot. The one in these figures is basically aligning this movement to another window of neural activity of the same length. We did another control where we use a method that generates data with the same statistics as the actual neural data. So it generates data with the same covariance across neurons over time and across conditions. And the lower bound is like 99% the same. We also did another control where we shuffle the movements, and the lower bound is a bit higher, but we are still like well above it. So yeah, all these controls. It will be on the paper. There's lots of the figures. Yes.
In this experiment, the movement of the tadpole in the screen is the same for both animals,
Yeah.
but in different orders.
Yeah. So
So
the question for people online is like if the movements are the same. So the targets are randomly generated, which is why there's like lots of movements. So each target in the sequence and each sequence is random. But since they were doing this task like for a couple hours, we could still match like enough movements to do the statistics. Also, like monkey C was much better at the task than monkey M. So, but yeah. This was one example, and this is like, I think, 11 comparisons across 3 different monkeys. So we could do this again across many monkeys. And another nice thing of this task is that we had many conditions, right? In the example that I showed you, there were 29 conditions. So different starting positions and reaching directions. So then we could ask, how is our ability to align the activity affected by the number of conditions? And remember that I told you that we think that the key is the similarity of the behavior, right? So according to our prediction, having many more conditions shouldn't make things much harder. So let's see what happens. If we only have a few conditions, we can align a bit better than if we have like 6 or 7. But after 6 or 7, things start like to plateau, right? This is each comparison, we are dropping conditions, and the black line is the mean across all of them. So if you look at the black line, you can see that it goes down, but after like 10 conditions or so, it plateaus. So if you're like into physics or extrapolation, you would argue that you can add more and more conditions, and this black line will still be very high, right? So our ability to align, even with this simple linear method, doesn't seem to be affected by adding more conditions. Okay. Yeah, now I really want to emphasize that this is not trivial, and really like behavioral similarity is key. So let me show you one more comparison that we did that we could perform thanks to what feels like an old paper now from my postdoc, where we were looking at neural activity across different tasks, which is something that people in the motor domain and the cognitive domain are interested in, right? Like what is similar and different in the brain when we do similar things, right? So for that, we look at 2 sets of tasks. The most similar one is monkeys using the same manipulandum to acquire the same 6 targets that you can see there in blue and red, only 1 dimension. And what we did was manipulate the manipulandum by forcing it. So then the monkeys would move the cursor just by generating force, or by letting the monkeys move the cursor, or by putting a spring that kind of pushes the monkeys back. So they have to basically push harder. Well, you know, this feels very similar, right? And actually, if you look, for example, at the velocity of the joystick, it looks very similar for these 2 tasks. So now the question, or the prediction, our prediction is that still these are not the same tasks. So the latent dynamics should be more similar across 2 monkeys doing the same task than the same monkey doing these 2 very similar tasks on the same day while we look at the same neurons. Okay? So what happens? This is 2 different monkeys. This is the data we've been looking at so far, like 2 different monkeys doing the same thing. And this is the same monkey doing 2 different but very similar tasks on the same day. So we are well below that. And the same is true for all the wrist tasks and another set of tasks where monkeys were reaching and grasping. So hopefully this convinces you that this preserved dynamics across monkeys really needs the same behavior. Yes, Tamar?
So this is a question from online, which I think you now just answered, but they're asking, is the movement alignment based on position data, trajectory only, or does it also include velocity?
So the activity of the alignment of neural dynamics is only based on being the same conditions, so the same targets, nothing else. It's completely unsupervised. Then this is why we have to do the relationship with behavior post hoc with decoders. Okay, any other questions? So I think hopefully you are convinced by now that, you know, we like actually 2 animals performing the same movement generate this similar dynamics in motor cortex. We were also curious if this is only motor cortex or any other brain region that is important for behavior. We also saw that, which is what we expected. So Junchal and Josh got recorded from dorsolateral striatum, which is putamen, so the sensory motor striatum in mice. And we could do the same thing. We could still align across mice, and the quality of the alignment was very similar to motor cortex and of the decoding. But now this is probably the most important result for a cognitive neuroscience unit, which is, what about things that are not moving, right? A lot of our life is a cognitive life, even the life of animals is a cognitive life. Like, for example, this is my dog judging me while I was teaching during COVID and not making a very good job at it. So, you know, what we did was we use a feature of the reaching task in which basically monkeys were presented with the target, but they had an instructed delay period, and they couldn't move during that period of time, but they are still preparing their movement, because when they get the go cue, they want to move quickly to get their juice. But we don't know that we know that they don't move, because if the cursor leaves the center of the screen, they get penalized, and they have to wait for a long time. So we're analyzing this period of time where the monkey is getting ready to move. And what happens is, as you would imagine, is that we can align the latent dynamics across monkeys when they are planning the same movements. And what is perhaps cooler is that we can build a model that predicts the future movement in one monkey, and it works equally well in a different monkey. So if you want, we can predict what monkey 2 is planning to do from what monkey 1 has done. Okay. Does this make sense? Okay, so to wrap up, I want to go back to the very beginning of this study, which I frame like in, the genome encodes instructions to build these circuits that have similar statistics across us, right? So this is why monkey brains are monkey brains and human brains are human brains. So how can we, and I want to show you that we do need the same brain architecture, probably, to find this aligned latent dynamics. So since we can't or wouldn't want to modify the circuit architecture of an animal, what we did was train neural networks to do this task. So we use a very simple method in which we train RNNs, and this is very common in movement neuroscience and non-cognitive neuroscience, to perform the monkey task. So you can see that we would give the RNN the target and ask it to produce the hand velocities that the monkeys produced. And this is very common in the field, as I said, so you can see the example output of one network. Now, our goal was to show that behavioral similarity is not sufficient. So what we did is we would use like this network as reference, and then we would train another network to perform the same task, but we will try to make the neural activity different from that of the reference network. And we did that by controlling a parameter that we call alpha. And you can see that we created networks with different levels of alpha, but their kinematics are still fairly similar. And for example, the correlation in the behavior across networks was very high. It's like around 0.90, regardless of the value of this alpha. So what happens now in the neural activity? If I compare standard networks, you can see that this correlation is super high. So all the standard networks find the same solution to do this task. But as I start increasing my alpha, I can make this activity more and more dissimilar until it actually becomes quite dissimilar. So just by tweaking this parameter, we can make very simple and arguably very stupid neural networks do this task by generating very different activity patterns. And what happens is, as we expected, is that they build different connectivity patterns. So these are two metrics of network connectivity. Basically, the dimensionality of the weight changes, dimensionality of the weights and the variance in weights. And you can see that these values are different for networks with different alphas. So this supports the idea that behavioral similarity is necessary for finding stable latent dynamics, but it's not sufficient, if that makes sense. Okay. Any questions? Yeah.
So this plot where you show the correlation as a function of the robots.
Mm-hmm. Yeah, let me find it again.
So you're showing similar plots for the data.
Yes.
But the data look more like your integrated alpha 0.5.
Yeah, that's a very good question. Like why?
So, but there we declare that it's
Yeah.
high correlation. Here, you're kind of bashing it as being, lousy networks that find
There's for people online, the question is why does alpha equal 0.5 look similar to the actual neural data, which is a very good question. So the answer is that we like thinking about motor cortex, or we used to like thinking about motor cortex as this part of the brain that controls muscles, and that's it, right? But in reality, in the last few years, people have found like attention signals. So the largest signal in motor cortex is very correlated to the diameter of the pupil. There's also reward signals. I will show you that there's also more cognitive signals in motor cortex. So we can match the kinematics very well, but we don't know anything about all these aspects of neural activity that we cannot match. Whereas in neural networks, these are like 300 neural networks or 500, and they only have to produce this activity pattern. So it's, in a way, this is why we need a very different upper bound and a different way to measure things. And actually, I think this has an implication, that is that if we try to do this alignment in prefrontal cortex in a complicated task, the animal is probably thinking about different things or has a different strategy. So the quality of the alignment will probably go down and will actually may reveal different interesting differences between animals in how they do cognitive tasks and the time course of learning. I hope this kind of gets it.
Just to double check, so when you build these graphs, are you subsampling the nodes that are in your model, like inevitably you're subsampling the brain?
Yeah.
Senses. So I can imagine these curves would go down subsample space.
Yeah, so the question is whether we are looking at all the units in the network or just subsampling. So we've done both, and they look very similar, because since these networks were only training these tasks, so basically all the neurons are working together and their activity is very low dimensional. Okay. Yeah, I guess like we have to start wrapping up. Tamar?
You have another 7 minutes.
Okay, so I can, I don't know what I can do in 7 minutes, but I guess I can introduce something that we're working on. I wanted to share with people here, that is, we've been studying like very simple tasks, right? Like center out, we don't do center out, right? Like a lot of our life is planning movements in an uncertain world. Like, and the example that my PhD student Bense, who's Bagge, who is the guy leading this, is tennis. Because, now that he's trying to hit the ball, and he has like some internal model of how a ball bounces, and he also knew like Djokovic prefers probably to hit it somewhere else, but still like he has to plan a movement without knowing the full information, right? And this is not what I've been talking about so far, and this is something that we've been working on for a while. So what Benz is doing in collaboration with Brian, who was a former colleague, is studying a task in which monkeys were given noise information about where their reward is going to be. So in the standard task, basically we draw a target around this circle, as I showed you, and then the monkeys have to reach that after some delay period. But in this uncertainty task, what we do is present a set of lines that indicate that whose centroid indicates where the monkey has to reach to get a reward. So you can see in the example at the top, the lines are very close together, so it's very easy, right? The center, where the center of these lines is. We can also make it harder by making the lines be farther apart. And then the monkey has to choose based on their best guess and move and try to get a reward. And the nice thing is that actually you can mess with the monkeys, because at the bottom, yeah, I shouldn't have said that probably live, but at the bottom you can see that even the targets were only on the top half of the screen, monkeys made very different reaches, right? But I want to show you, and I have very little time, that actually monkeys learn how to do this task and take the full information available to them into consideration when planning movements, as humans do when they do this task. So I'm plotting here where the monkeys reached compared to the centroid of these QCs. So if the monkeys were perfect at the task, if this plot should look like this identity line, right? If they were ignoring this information, the smart thing for them to do is like move to the center, because this is the safest bet. So they will be doing this. So let's see what happens. In the low uncertainty trial, so when the lines are very close together, the monkeys are very good. So you can see that the actual reaches during one day in orange match perfectly the ideal, basically the optimal observer. But if the lines are very far apart, what the monkeys do actually is they play it a bit safe and they try to reach more towards the middle than they should. Because they know that things mostly happen in the middle of the workspace. So I would when in doubt, I would reach them. Okay. So monkeys were actually pretty clever about this. So and we wanted to know how they do it. And I don't know what to tell you in 5 minutes, I guess, other than we recorded from the key motor regions for movement planning and execution. So primary motor cortex is the main output that I was telling you about. Premotor cortex is more interesting in the sense that it's upstream. It integrates information from prefrontal cortex, cerebellum. So there's more like if you want, cognitive stuff going on there. And we recorded from hundreds of neurons from these regions, or Brian did. And just like, and we focus again on movement planning. So after the target is presented and before the monkey moves. So I guess very quickly in one, I'll go through 2 results only. One is that we were wondering if monkeys somehow think that these are 2 different tasks. So you would think that in prefrontal cortex, this is a very different task. So you would pick large differences in neural activity, right? Because one is like dumb and straightforward and the other is more complicated. The question was, can we also pick this difference in motor cortex? And to our surprise, we found that the manifolds for these 2 tasks are very similar, but we could classify which of the 2 tasks the monkey was performing even before the cues were presented. And this is because the monkeys did the tasking in blocks. So somehow the monkeys knew like, oh, I'm now in this uncertainty mode, so I have to plan things differently than if I'm in this like very straightforward mode, which we were not expected to find in primary motor cortex. But I guess the main result here, the main result is looking at how activity is organized in these regions. So what I'm going to show you, these are the first 2 dimensions of the manifold, and it's like one session from a monkey. If you look at premotor cortex and plot each trial as a single dot, you see that orange dots are in one place and blue dots are in a different place. So this is quite unexpected for motor people like Tamara and me, because premotor cortex is the motor planning region per excellence, but activity is dominated by whether the lines are close together or not. And if actually you color code these dots by reaching direction, is a complete mess. Whereas if you look at motor cortex, if you color code by uncertainty level, everything is mixed together, but we can clearly see a gradient based on where the monkey is reaching. So these 2 regions that are, again, next to each other in the brain, so remarkably different activity patterns as soon as you make the behavior a bit more interesting than we motor people are used to. This is, but how long do I have to?
You have to.
Okay, just very quickly, despite these differences, if you look at the entire 10 dimensions of the manifold, you can decode both uncertainty and the reach direction from both regions. And reassuringly, you can decode it earlier on from premotor cortex, which is, earlier on in the neural axis. And I guess in the interest of time, I will just tell you that the activity within the manifold looks very different. Oops. So of course this is not working. Just to reinforce this message, of course this is not working. Oh, beautiful. Let me just do this. Okay, I just skip this. Move to the final slide. Anyway, I've told you very quickly that premotor cortex is mostly about uncertainty. Motor cortex is mostly about movement. But this information is in both if you look at enough dimensions in the manifold. One thing that I didn't have time to show you is that this information lives in 2 orthogonal dimensions. So in theory, you could move along the uncertainty dimension, right, without affecting the movement dimension, which is convenient because then you can plan this movement the same way, even regardless of how the information is presented. And this is all very reassuring, but we are wondering, are actually these 2 signals used combined in motor cortex to plan the movement? Or is just like this visual uncertainty something that we see just because the monkey sees it, and we know that these regions have input from visual regions, no? So the best we could do was like come up with, again, a neural network model that we fitted on neural data. So we present the same inputs as the monkey saw, and we have a PMD and an M1 module, and we train it to reproduce the data that we record from on each trial. And we also are able to, this basically give us to have a model of the brain, of the neurons we recorded from, and then we can manipulate the inputs, present different inputs, and see what happens. Okay, just to show you, the RNN looks like the brain, so premotor cortex is mostly about uncertainty, and motor cortex is mostly about movement. But the cool thing is that we can show a different set of inputs and play with the uncertainty of these inputs, and we can see a bias that represents how the the monkey would be behaving, which indicates, and sorry because this is a bit too fast, that actually these two signals are combined in the motor cortices to guide movement. It's not that the visual information is only there as a byproduct. And I'm happy to talk more about that. Now we'll just like wrap it up. Oh, we also found this in mice, in mouse data, where we could also look at prefrontal cortex, and prefrontal cortex is more cognitive, as you could expect. So about running a bit too long. So to wrap up, I told you about this idea of neural manifolds as a theory of how the brain works. I presented this paper on bioRxiv and now in Press about how different animals performing the same covert or overt behavior generate similar dynamics. I very quickly tried to summarize ongoing context that I love because it starts to have more cognitive aspects into movement. And I didn't have time to tell you about our study of neural manifold geometry, but this is on bioRxiv, so you can have a look and email me if you're curious. So I guess I should thank you for having me. It was a pleasure, but even if the circumstances were not the best for having to fill in Sliman's shoes, I'd like to thank people who did the job, the work, those involved mostly, and we'll be looking for PhD students in the fall, so tell your friends and next year. And yeah, thanks for your questions and your attention.
If it's okay, we're going to start with a few questions from the audience online. So there's a few written ones. Feel free to speak up if you prefer. But the first one is, what dimensionality reduction techniques have you used or prefer to obtain the neural models?
Yeah, very good question. So we like to keep things simple as far as we can. So we use principal component analysis a lot because it tells you what are the patterns of activity that dominate what this population of neurons is doing, which seems reasonable. In this paper that I couldn't talk about, we actually test how well like these linear methods work. So I don't know if you can see my hand, but if you imagine the manifold is like a ball like this, and if you only explore like a little bit, then a plane is a good approximation. But if you go very far, then a plane becomes pretty bad. So in that paper, we show that you may need to use linear methods like isomap or umap when behaviors become more complex or for certain brain regions, but it's a bit too long to describe. But if you go to Google, to bioRxiv and look up Katia Fortunato, she's the first author, and you can see a lot there on that.
Okay. Andy, do you want to ask your question? Because it's long and complex.
Hi. Yeah, so thanks for a great talk. Really interesting stuff. I hope you can hear me. So it was really in regards to whether you could show that the behavioral similarity really is insufficient to explain the similarity between latent dynamics. So it struck me that in the data you showed of your model, you for your different networks, you did have a strong correlation in the first two modes, right?
Mm-hmm.
And you trained your models to reproduce two-dimensional XY position data. So presumably you would say, well, I'm going to get similarity in those two modes because those are reflecting the XY position data. And it just struck me that in a natural movement, you've got all sorts of biomechanical constraints that are going to lead to similarities in, I would have thought, higher than 2 dimensions. So just as an example of an additional dimension that is probably similar between monkeys, there'll be things like limb stiffness might be modulated through the movement. So I wondered if you sort of train the model to actually control a biomechanically realistic arm, whether you wouldn't see constraints acting across more dimensions, and then you would then see similarities in the modes across more dimensions that would be more similar to what you see in reality, if that makes sense.
Yeah, no, that's a really nice question, and we've been thinking about this. And yeah, you're right. So the 2 dimensions that we can align perfectly, I didn't want to get into detail, but it's because our RNN is producing the output through a linear readout. So since the network has to produce the same signals through a linear readout, and our method aligns things that are linearly alignable, so this is why we get the perfect correlation. For the things like, we think that the spinal cord is very nonlinear, right? So I would expect that in an actual animal where we have, multiple descending systems acting on these very nonlinear spinal cord circuits that then activate motor neurons through these complicated biomechanical constraints, I would think that correlations identified with a simple linear method would be lower. But I agree with you that it's an interesting problem. And this is why we're putting some effort into training ARM models, by the way. There was something else I wanted to say that kind of slipped my mind because I'm a bit tired, but I'm sick. But yeah, Andy, any thoughts on that?
Let's sorry, Andy.
No, you carry on.
Thank you. Questions from the audience in here. Katie?
I'm wondering whether when you build an RNN or through your data, does it make predictions about when it would be not on the same manifold? Like, is it about the visual stimulus? Like, for example, like spontaneous movements, would that be on?
Yeah, there's for people online, like whether we can use RNNs or the data to predict that things would be out of manifold. So for RNNs, Joanna, who is here, Chang, we have a paper on bioRxiv with Claudia Klopath, where actually this was something that we realized only in that study, which is where this study is about how the repertoire of a network shapes subsequent learning. And what we realize is that just by presenting the inputs in a different way, even if the output is the same, you can be completely different representations in neural space in this network. And the simple example was if instead of indicating the network, you have to reach to an angle of 0 degrees, 30, 60, so forth, you tell the network, I'm going to call this reaching goal 1, this 2, this 3, sorry, and this 4. Then the network doesn't kind of build a good representation. That is, if you specify the continuous angle, you build this manifold where similar movements are next to each other, like in the brain, right? Like these trajectories were next to each other. If you use arbitrary inputs, then the network doesn't know how to make sense of them. You would like this as someone interested in semantics. So then the different representations don't follow any logical organization. So just by using different inputs, you are creating a very different manifold that is much more complex and it's not useful for adapting. So I think this is a very important observation, like for BCIs, which is related to closeNIT. If we do like curriculum learning training and we want to develop a training paradigm, we have to be very careful of each step. And given the nature of the inputs or the information is more important than at least I used to think about.
Fantastic, thank you so much.
Thank you.
I'm slightly excited. I'm slightly excited because there's some parallel ideas that we're playing around with semantics, semantic representation of being manifold.
Oh, that's super cool.
So we're fitting neural data as a manifold to semantic representation space. Slightly different, but I think conceptually it's not. So I was curious about a couple of technical things. So how many neurons do you need to sample before you get a stable manifold? How do you deal with off-target measuring? Because you're plunging some electrodes in, presumably miss sometimes. Yeah. That's going to get more complicated when you're outside of nicely localized functions in the brain. Yeah. And then I was also wondering whether in the manifold, whether the dimensions are interpretable. So we've been thinking about this a lot in semantics. So you can imagine the internal space which you can't give an interpretation to, right? You can't necessarily line with a semantic feature, but you can still be coding semantic similarity structures. So I was sort of wondering whether those manifolds, the dimensions of the manifolds are actually interpretable, perhaps to do with they're in space or certain muscle connections have to be meaningful. Yeah, if that makes sense.
Yeah, no, it does make a lot of sense. I'll try to rephrase.
How many neurons?
Yeah, no, I think So how many neurons? This depends on the brain region and the task. So for the kind of central tasks I've been talking about, 20-ish is enough. And it doesn't matter, we get the same results if we basically drop all the tuned neurons. If you have 20 untuned neurons in the classic way, you get the same results. As the task becomes more complicated, you may need more neurons, but even for the whack-a-mole task, 25, 30 was enough. This is for motor cortex. I'm collaborating with a hippocampal group, and there where the neurons fire so sparsely, like in CA1, like places fire like very little, then you need many more because otherwise you don't have any spiking. This was one. What was the next question?
So how would you deal with sort of like on-target hits?
Yeah. Whether it really matters if we're recording from the right spot, I guess. I think for motor cortex, we are surprised because in one of these studies, we were using a monkey with an implant in the arm area to decode hand kinematics and hand EMGs, and it was like 90% as good as in the proper monkeys. And there's a paper, for example, showing from the Chenet group, another great scientist that we lost recently, by the way, where they show that you can decode speech from arm motor cortex in people with brain implants. So I think the information may be available for decoding more, in more regions than in the regions that actually do the doing. And my intuition is that it seems to be the case that it's hard to get rid of these signals. Maybe they're not used for anything, but they are there. But I don't know. And I don't know enough about language-related regions, but it sounds like we should have a chat about these things. And what was the...
Once you've got your manifold component, whether those components are interpretable.
Yeah, the last question is whether the dimensions of the manifold are interpretable. And at the beginning, Juan in 2016 was really hoping that, or 2015, that we will find dimensions that are the extensors of the hand, flexors of the hand, and it's not the case. What seems to be the case, for example, in uncertainty data, I couldn't show it, but you kind of show it a bit that in premotor cortex, just looking at what dominates activity tells you that it's uncertainty, and motor cortex is movement. So I think these kind of things can be done, but then this is why you always have to add like a decoder or some other kind of readout to try to make sense of the data. So what we usually do is we have our hyperplane with PCA, we build a decoder, this gives us a direction. And then you can ask what happens in the other directions? Or are there many directions that have the same information and so forth?
A quick question, follow-up questions related to that from online is whether you pre-select your neurons.
Yeah, no, we don't pre-select our neurons. As I said, we've even gone as far as dropping all neurons that are tuned to movement in motor cortex to repeat the analysis. And I think in hippocampus also dropping all the places, and you can still represent the manifold. I think it was. Yeah.
You were mentioning skeptics.
Yeah, thank you.
This follows from a very general principle that the activity in the neural system is going to be more similar for more simple things outside the world, which will follow, for example, if a neuron's got a preference for a certain direction of reach or a goal or whatever. Because it seems to me like if you go back to your first experiment, you're going to plot 8 target locations in a 2-dimensional space. That's already extremely constrained what can happen, which is that you're going to end up with a circle.
Yeah.
Is there something more than that just broad idea that similar things are similar in the brain?
Yeah, I think that is a fair question. I mean, it is a fair question, a good question. And what we're thinking about this is, we again have this genome that and our developmental process, right? But there actually can be 2 options, right? We have all the biomechanical constraints that Andy was talking about on Zoom. So maybe through our developmental and learning experience, we developed 2 slightly different solutions to achieve the same problem or, reaching to my bottle of water. So what this shows is that actually there's more to it than that. I also think this study is the beginning because we are, for example, interested in seeing how this applies to cognitive tasks and try to identify differences between animals solving the same task with different strategies or different training histories. We're also interested in how models of neurological diseases that have very subtle changes in neural activity change their manifold, and what cannot be aligned may be a good descriptor of what is different in their brain at this systems level. So I think I kind of whistled my way out of your question, but I think this is only the beginning of doing comparative experiments within a species or even across species of how they solve similar problems.
I think it's a great place to stop. So maybe we take a question after releasing the audience. Before I thank Juan, I just want to say he's a good friend, a collaborator. We can bring him over here as often as you guys want to interact with him. So please reach out to me or Juan if you want to follow up on the chat. So thank you so much, Juan.
No, thanks to you.